The overall goal of this image analysis pipeline is to perform quantitative volumetric analysis of T1-weighted MR images and to identify critical change points when brain atrophy begins in neurodegenerative diseases.
This method can help answer key questions in quantitative analysis of brain anatomical changes during the course of neurodegenerative disease. For example, the order and pattern of brain atrophy in the Huntington's disease. The main advantage of this technique is that the combination of atlas-based segmentation and change point analysis bring unique spatial-temporal information about brain degeneration from large populational studies.
The volumetric analysis of brain MRI was achieved by MRI Cloud, which provide fully automated image segmentation and quantification through remote computation and web-based user interface.
Change point analysis of the brain MRI provides us a systematic view of the disease progression across the whole brain. The applications of this technique extend toward a range of neurodegenerative disease.
To begin this procedure, convert three-dimensional T1-weighted images from vendor-specific DICOM format to analyzed format. Double-click to open DCM2Analyze.exe. A pop-up window will open. Specify the input DICOM data directory path as input and analyzed image file path and name as output. Then